Very good morning, my dear students. Today we are going to see about transmission electron microscopy. What is an electron microscope? Usually, electron microscopes are scientific instruments that use a beam of highly energetic electrons to examine objects on a very fine scale. This examination can yield information about the topography, morphology, composition, and crystallographic information. And it has two types. One is transmission electron microscope, another one is scanning electron microscope. The TEM allows us to study the inner structure of a specimen or a material, etc. The SEM is used to visualize the surface of objects. Next, we will see about the principle of a TEM. We all know electrons possess a wave-like character, and the electrons emitted into vacuum from a heated filament with increased accelerating potential will have small wavelength. Then such higher energy electrons can penetrate distances of several microns into a solid. If these transmitted electrons could be focused images with much better resolution. Focusing relies on the fact that electrons also behave as negatively charged particles and are therefore deflected by electric or magnetic fields. And we can see the parts of a TEM. They are condenser aperture, objective aperture, specimen hold port, selected area aperture, screen under cover. This is the cross-sectional view of a TEM. In this, there are four important parts. The first one is electron source. The electron source consists of a cathode and an anode. The cathode is a tungsten filament which emits electrons when being heated. A negative cap confines the electrons into a loosely focused beam. The beam is then accelerated towards a specimen by the positive anode. And the second system is electromagnetic lens system. The electron beam is tightly focused using electromagnetic lens and metal apertures. And the third one is sample holder system, a platform equipped with a mechanical arm for holding the specimen and controlling its position. Finally, there is an imaging system which consists of a phosphorescent screen. Now, we will see about the working of a TEM. We know the specimen is bombarded by a beam of electrons, which are primary electrons. No, the bombarding electrons are focused to a bundle onto the object. In areas in the objects where these electrons encounter atoms with a heavy atomic nucleus, they rebound. In regions where the material consists of lighter atoms, the electrons are able to pass through. The fine pattern of electrons leaving the object reaches the objective lens, forms the image. It is then greatly enlarged the projector lens. Eventually, the traversing electrons reach the scintillator plate at the base of the column of the microscope. The scintillator contains phosphor compound that can absorb the energy of the striking electrons and convert it into light flashes. Thus, a contrasted image is formed on this plate. Here, these are the images of TEM, Golgi complex, and a TEM image of a mitochondria. Then, there are some more advantages as well as disadvantages for TEM. First, we see about the advantages. TEMs offer very powerful magnification and resolution. TEMs have a wide range of application and can be utilized in a variety of scientific, educational, and industrial fields. It provides information on element and compound structure. Then the images are high quality and detailed. Then the disadvantages are TEMs are large and very expensive, and it needs laborious sample preparation and operation and analysis requires special training also. And samples are limited to those that are electron transparent. TEMs require special holding and maintenance. Images are black and white only. We cannot obtain the color images out of TEM. Then there are some more biological applications of TEM. You see, in medicine as a diagnostic tool, it is very important in renal biopsies, and in cellular tomography also it plays a major role. We know the tomography refers to imaging by sectioning through the use of any kind of penetrating wave. Information is collected and used to assemble a three-dimensional image of a target. This is used for obtaining detailed 3D structures of subcellular macromolecular objects. And in cancer research also, it gives its contribution. It is used to study the tumor cells ultrastructure. And in toxicology also, this is used to study the impacts of environmental pollutions on the different level of biological organizations. Dear students, today we have learnt about the principal parts and cross-sectional view and working, advantages and disadvantages and biological applications of a TEM. Thank you.